Welcome to today's lecture on advanced topics in microbiology. In this session, we will delve into various aspects of vector-borne diseases and their interactions with their respective vectors. We will explore the immunopathogenesis of malaria, the pharmacogenetics of antimalarial drugs, the mechanisms of vector-parasite coevolution, and much more. So let's begin. Trypanosomiasis, caused by the parasite trypanosoma, is a significant health concern in many parts of the world. The interaction between trypanosoma and its vector, the tsetse fly, plays a crucial role in disease transmission. The parasite undergoes complex developmental stages within the tsetse fly, influencing its vector competence and ensuring survival. We will discuss these interactions in detail, including the molecular mechanisms involved and the potential targets for vector control strategies. Malaria, caused by Plasmodium parasites, involves a complex interaction between the parasite and the host's immune system. We will explore how the parasite evades the immune response and the factors affecting disease severity. This knowledge is crucial for developing vaccines and treatments. The efficacy and safety of antimalarial drugs can vary among individuals due to genetic factors. Pharmacogenetics, a field that studies how genetic variations influence drug response, can provide crucial insights into the optimization of antimalarial therapy. We will discuss the genetic variants associated with drug metabolism, drug-target interactions, and individual susceptibility to adverse drug reactions. This knowledge can guide personalized treatment approaches and the development of novel antimalarial drugs. Vector-borne diseases exhibit complex host-vector-parasite dynamics, including coevolutionary relationships. The coevolution of vectors and parasites involves reciprocal adaptations, genetic changes, and selective pressures that shape vector competence and disease transmission. We will explore the mechanisms governing these coevolutionary processes, including vector immune responses, parasite manipulation of the vector, and the impact of environmental factors. Understanding these mechanisms is crucial for predicting disease dynamics and designing effective control strategies. Leishmaniasis, transmitted by sandflies, poses a significant public health threat in many regions. Vector control strategies play a key role in reducing disease transmission. We will discuss various approaches, such as insecticide-based interventions, environmental modifications, and the use of genetically modified vectors. Additionally, we will explore the challenges associated with vector control, including insecticide resistance and community engagement. Filariasis, caused by filarial nematodes, affects millions of individuals worldwide. Molecular diagnosis techniques have revolutionized the detection and characterization of filarial infections. We will delve into the principles of molecular diagnostics, including PCR-based methods, DNA sequencing, and antigen detection assays. The advancements in molecular techniques allow for accurate and early diagnosis, allowing timely intervention and control measures. Apart from vector and parasite interactions, abiotic factors significantly influence disease transmission dynamics. The interplay of temperature, humidity, rainfall, and other environmental factors can impact vector abundance, survival, and vector-borne disease prevalence. We will explore the influence of these abiotic factors on disease transmission dynamics, considering both short-term and long-term effects. Understanding these factors aids in predicting disease outbreaks and designing effective control strategies. Babesiosis, caused by the parasite Babesia, is a growing concern due to the emergence of drug resistance. Understanding the mechanisms of drug resistance in Babesia parasites is vital for developing effective therapeutic interventions. We will discuss the genetic mechanisms underlying drug resistance, including drug efflux pumps, target site mutations, and gene amplifications. Additionally, we will explore potential strategies to combat drug-resistant babesiosis, such as combination therapy and the development of novel antiparasitic agents. Chagas disease, caused by the parasite Trypanosoma cruzi, poses a significant health burden, particularly in rural communities. Understanding the epidemiology of Chagas disease, including the patterns of transmission, reservoir hosts, and risk factors, is essential for effective prevention and control strategies. We will explore the various epidemiological aspects and discuss the challenges associated with disease surveillance, early detection, and community education. The variation in host susceptibility to vector-borne diseases can be attributed, to a large extent, to host genetic factors. Genetic polymorphisms in host immune response genes, receptor genes, and other key molecules can influence disease susceptibility and severity. We will examine various host genetic factors associated with susceptibility to vector-borne parasites, including genetic association studies, twin studies, and functional genomics approaches. Understanding host genetic susceptibility aids in identifying at-risk populations and developing targeted interventions. The bacteria in mosquito guts can either help or hinder the transmission of dengue virus. We'll learn how these bacteria affect dengue transmission and how this knowledge could help us develop new ways to control the mosquitoes and prevent the disease. Bancroftiasis, caused by the parasitic filarial worm Bancroftia, poses diagnostic and therapeutic challenges due to its complex life cycle and elusive clinical presentation. We will discuss the current approaches and emerging technologies in the diagnosis of Bancroftiasis, including serological assays, imaging techniques, and molecular diagnostics. Moreover, we will explore the treatment options, challenges in drug development, and strategies for effective patient management. In summary, vector-borne diseases are a major global health concern. Understanding and controlling these diseases requires a multidisciplinary approach. We have covered diverse aspects, including parasite-vector interactions, molecular diagnostics, and disease control strategies. Medical professionals must stay informed to address this burden.